So, hi everyone, my name's Arlie, my pronouns are they, them, and she, her. Before starting the presentation, I'd also like to extend my acknowledgement and respect to the custodians of the land on which I'm zooming in from, the Yagara and Turrbal people of Meanjin. Paying my respects to their elders past and present. I'd also like to draw attention to the fact that next week there's Trans Visibility Week. I'm a fairly visible trans person, but there are a lot of trans people who aren't visible in that same way. And so, yeah, Trans Visibility Week is a nice time to sort of acknowledge the sort of broad range of trans people who exist. It's quite likely that you know someone who's trans who doesn't have the safety or security to be out. So taking the time to show your support can be a nice way to let people know that they're safe with you. I think as well with the election news last night, it's a pretty sad time for a lot of people in the queer community. So using it as an opportunity to get informed on issues if you don't quite understand things, it's a great time to do it. Yeah, and if you have any questions on any of that stuff, feel free to reach out to me whenever. Right, so on to the presentation. So today I'm talking about PhyloG2P, which is a sort of emerging area of research, which is using phylogenetic methods to map traits to genes. So I'm doing a review paper on this at the moment, along with my supervisors. And so the sort of purpose of this talk is to cover some of the sort of, underlying theory and what it's about, and look at how it can be applied and why it might be relevant to the center. So in the talk, I'm going to start by just sort of working out where it fits in relation to the sort of existing G2P analysis like QTL and GWAS stuff. I'll then give a bit of an introduction as to how the methods work, including some discussion of replication, which is very relevant to these methods. We'll then look through some of the tools that are currently available to do this analysis, and I'll sort of conclude by giving a overview of what the process of applying this analysis to your sort of, species and data that you have would look like. So beginning with the sort of current, like how do we know what genes do? We sort of have the option of mutant libraries with everyone's favorite weed here. And so if we have a particular trait that we're interested in, we can look at a large number of mutants and find variation within that trait and associate that with the particular changes in genes. Another approach is using QTL analysis, where we find individuals from a population that are contrasting in a phenotype we're interested in, and then we can do a whole bunch of laboratory crosses with them and look for associations between the trait and particular SNPs. And also we can look at GWAS, where we might take like an existing population and then we look for correlations between the presence and absence of particular SNPs and the trait values that we're interested in. So these analyses have some limitations. So the sort of big mutant libraries only really exist for model organisms, and it can be quite costly to set up, particularly if you're sort of interested in a lot of genes or a lot of traits. For the QTL analysis, your species need to be able to grow in the lab. So if they've got long generation times or they're difficult to cross in a lab, then this analysis is going to be very tricky. If anybody was watching David's presentation on GWAS analysis last week, they'll know that population structure can be a bit of a challenge for GWAS. And this is really exacerbated when we start talking about like sort of distantly related species. And so if the traits that we're interested in understanding only show significant variation between species, then these methods aren't really going to work. And particularly if we're looking outside of model organisms, then there's limited options with these to be able to understand the full breadth of traits and how they're controlled. So what can we do about this? Well, this group of researchers here, they sort of put their money behind phylogenetics being the solution. They released this review paper a few years ago that has a very sort of bold title. I think I would partially agree with it at this stage, and hopefully by the end of this talk, you'll understand why they believe this might be the approach that we'll be using in the future, and you can make your mind up as to how much you agree with them. But the basic principle that they put forwards is that the population genetics methods that we're looking at in the last couple of slides really focus on like individual species or maybe recently diverged ecotypes or species. But if we sort of take a phylogenetic approach, we can actually include a much broader range of species, and we can use the long history of evolution that has led to those species to help us understand the relationship between traits and genes. So what does this sort of look like in practice? So say if we wanted to find a frost tolerance related gene that we could introduce into domestic crops, then maybe we would look to wild crop relatives that have frost resistance. And so then we could look at sort of another wild relative that doesn't have it. And if we sort of got their genomes together and we had a look for places where they're different, we could see, okay, maybe there's a spot here that's different between the two. So we could say, well, cool, this is related to frost tolerance. But often it's not actually that easy, because if we're looking between distantly related species rather than different individuals within one population, then it's likely that there's a lot of other differences between them too. So in this case, there might be sort of, high salt content in the soils where this species grows. And so there might be some other genetic changes associated with salinity. And likewise, this species here, maybe it's got a unique pest that's a problem for it. And so it's developed resistance to that. And so without knowing which of these genes is the one that's related to frost tolerance when we're looking between the two species, we're at a bit of an impasse, which brings us to the first challenge. So for all of these different genes that we have, how do we know which ones are actually the ones that we're interested in when there's sort of so much variation between these two species? So the answer to this that is used by PhyloG2P methods is replication. So replicated evolution refers to any time that like distantly or unrelated organisms have independently evolved similar solutions to similar environmental challenges. So on the left here, there's an example with pitcher plants where all of these look very similar, but they've come from completely unrelated lineages and evolved completely independently. And likewise, human domestication on crops has sort of imposed this repeated evolutionary sort of like selective force. And we have sort of independently ended up with a lot of species that have increased sort of grain counts and shatter-resistant genes and all the other properties that we want for crops. And so applying replication can allow us to, like, rather than looking at the contrast between sort of one species with frost tolerance and one that doesn't, we could instead look at multiple species, which we'll refer to here as like the foreground, which all have frost tolerance, which are these sort of species that are marked in blue here at the tip of the phylogeny. And we can contrast those with a whole bunch of ones that are not frost tolerant that are in the green here. And so with sort of, when we see these species that are frost tolerant, when most of them are not frost tolerant, we might suspect that sort of, this frost tolerance has arisen independently multiple times. And that then somewhere along sort of the evolution that's occurred on this branch, there has been some change to a gene that allows it to survive in these colder climates. And so now when we look across the whole genome, we can try and find these genes that are like occurring repeatedly for the frost tolerant ones and not in the ones that aren't frost tolerant, and filter those out from the red ones that are unrelated traits. So this brings us to a second challenge. What are we actually looking for with these genetic differences? So there I had these sort of little blue bars to say that somewhere in the genome there's something going on, but what are we actually looking for? If we want to build a model, we need to know specifics. So there's a few different ways that that sort of similar replication in a trait can occur. So we might have, in the most fundamental case, an identical mutation. So if we have a specific coding region for a protein, and that protein gives some sort of antifreeze properties or something like that, then maybe having specific substitutions in that protein allows it to work more efficiently or something. But here we'd have some ancestral state where we've got this ATGA, and then in the replicate, that T has been subbed out for a C in the species that we're looking at. On a broader scale, we could have similar genes, but different mutations that are taking place. So again, maybe there's a gene that's related in some way that can sort of infer these properties of frost resistance, but maybe there's multiple changes to that gene that could result in that frost tolerance. And so we might see in some species that have taken one path, whereas in other species that have taken another path. Particularly for traits that are a bit more complex, we might actually see that there's completely different genes involved in being able to provide that tolerance. And so in this case, we've got a change in gene 2 that might give us frost tolerance, whereas here it's a change in gene 4. And so the PhyloG2P methods typically focus on the types of changes where we're seeing things occur in common genes, although there are some methods that can factor in to try and identify cases where we have got different genes within the same pathway. That's probably a bit too complex to get into in this discussion, but if anyone is interested in that, they can follow up with me later and I can provide some insight. But so the other question that comes up is what actually is replication in this case, and like in which sort of traits and in what kind of circumstances do we expect to see the kinds of genetic changes where we could identify one particular gene as being involved in the replicated evolution of the trait. So the way that we understand traits and species, there's a lot of different scales to it. We could have simple protein functions like I was talking about with that antifreeze, where maybe there's little changes in pink here that give it the capacity to bind to the screen star when it used to only bind to the blue square. If we looked at a cellular level trait, we might see sort of new cellular structures like a trichome appearing. And so then, there might be one particular gene that is highly involved in creating these, or maybe there's multiple genes involved. We can scale up to traits as well that are maybe on the more macro scale. And so we could have something like stem size in these mushrooms here. And so like with anything that is sort of continuous, there's the chance that it's actually, lots of loci of small effect. And so that might make it more difficult to identify the common genes, but we'll talk a bit more about continuous stuff later. And then we can also look at really complicated phenotypes. So something here like this ant-eating phenotype with the generic mammal that maybe looks a bit like a dinosaur, not being able to eat ants. Whereas then we have the echidna and the anteater that both evolved these similar long mouths with a long tongue that's specialized for eating ants. And we can think about the trait of sort of ant-eating here as actually being composed of multiple other traits on these sort of lower levels, like the tongues end up being sticky, so maybe that's more of a cellular trait. They're longer, so we could break that down into a sort of macro trait. And it's possible that we might see similar changes in proteins as well. And so whenever we're looking at these sort of layers of traits that are replicated, we can often break that down to simpler levels, and sometimes that'll help us work out whether they're really replicated or not. So bringing this together to give an example of how PhyloG2P analysis works, what we're doing with this analysis is we'll take sort of a large alignment of genes or a whole genome in an alignment, and we'll go through that finding candidate genes sort of that are aligned between all of our species. So in this case, we have a group of species where we've got the sort of tall standing form trees of eucalypt as well as the malleeform trees. And so the malleeform has evolved multiple times independently. And so we might want to understand what's driving this particular form of the trait where, they have this short form. And so then what we're really looking for is like candidate genes where they match a particular pattern, where we're seeing that on these branches where there's been this sort of, at the end of this evolution, we've got the mallee tree, we're looking for some change that's happening consistently there. So maybe that's the identical mutations, maybe it's different mutations in the same gene, but we're looking for genes that fit that pattern effectively, where there's something happening for all these blue branches and it's not happening on the green background branches that don't lead to the mallee tree. In contrast, if we saw that there was a change that happened on one of these blue branches, but also happened on two of the green ones, then we wouldn't necessarily think that that has anything to do with the malleeform. It may, but chances are it doesn't. So talking about some of the specific applications that exist for these methods, looking at the identical amino acid, identical substitutions case here, I've got an example with Rubisco and C3 and C4 plants. So the C4 photosynthesis has evolved multiple times independently, and so we've got it on these blue lineages here. And if we look at the Rubisco amino acid alignment for C3 plants at this particular residue, they've almost all got an A, whereas for C4 plants have almost all got an S. And so if we were able to scan through the whole genome and we didn't know that Rubisco was related to C4 photosynthesis, then methods that can detect those identical amino acid substitutions would be able to flag that Rubisco is important to the trait that we're interested in. So there's recently been a couple of web applications released that can do this. So you can just take sequence alignments that you've got and trait data and upload those onto these web browsers and the web servers, and they will run this analysis for you, which makes it very convenient. But there's sort of a limitation in that it only detects identical substitutions, which whilst that is the purpose, there's a lot of evidence that in a lot of contexts that replicated evolution can occur through different changes in the same gene, or it might even be that we have a change in a particular amino acid residue, but that it actually changes to multiple different amino acids that sort of give the same function within the context. So these methods are sort of very quick to run, very accessible, but there's a lot of context where they might not be able to identify results. So cases where you know that it's something to do with specific changes to proteins, these are pretty good. But if you suspect the trait might be a bit more complicated and is maybe controlled by non-coding regulatory elements, then this might not be the best approach. Instead, you may be interested in looking at the changes in substitution rate. So if we're evolving a new trait, we expect that there's got to be some change in the underlying genome to sort of change the phenotype and to give us the different form of that trait. And so these methods try and identify that as seeing an acceleration in the rate at which mutations are being fixed in the population. So in this example here, we've got wet adapted eucalyptus and the multiple transitions into dry environments. So we've been looking at this in a group at UTAS with the Ute drought experiment, and we're planning to use PhyloG2P analysis in that system. And so in this case, we might see that in the background species, they're, sort of that have all been in these more wet environments, that they haven't had much change to their genome necessarily, where there's been, like, for a particular gene we're interested in, it might be that it stays pretty consistent. But then for all of the dry adapted species, we might see that they undergo a bunch of changes as they change the function of that gene so that they can get that resistance to the drier, hotter environments. We might also see the inverse of this, where maybe there's a gene that's really essential for drought tolerance that we see a reduced rate of mutation in, whereas the background where there's plenty of water, it doesn't matter if there's changes to that. So maybe a trait that controls sort of, when the stomata choose to open and close, where if you sort of mess around with that when you've got very small margin for error in a dry environment, that will kill you, whereas if you're in a wet environment, that's not such a big problem. Sorry. So these two groups here, the PhyloAc and RER Converge, are sort of two approaches to doing this analysis that they sort of require a bit more independent running than the web applications from before, but they have good walkthroughs for both of them. Another interesting area that's really opening up in the past few years in PhyloG2P analysis is looking at continuous traits. So in the context of replication, continuous traits are a bit different, where instead of saying that we sort of got some species that do have the thing and some that don't, we're instead looking for replicated changes. So here we've sort of, got some species that have got lighter flowers and darker flowers, and then some that have got lighter and darker, darker, lighter, darker. And so we're looking for these sort of like replicated changes in phenotype over the phylogeny. And so the full details of how these continuous methods work is a bit complicated, and again, beyond the scope of the time I've got for this webinar. But the basic idea is that we're looking for rates of genetic evolution, so like that number of substitutions we're looking at on the last slide, and we're looking to see if there's any relationship with that and sort of changes in the phenotype. So if we have a very small change in phenotype, like where sort of, these two flowers here are probably quite similar to their common ancestor, given that they're similar to each other, then we'd expect to see a small change like in phenotype and a small change in rate. Whereas between this like very dark flower here and the lighter flower, there's likely been several genetic changes that have led to the difference in pigmentation. And so where we've got this larger change in phenotype, we might see a larger rate of evolution, but we may also see different relationships. And yeah, again, the full details of that are a bit beyond the time that I've got. But if you've got continuous trait data, there's some really good evidence that you can get a lot of information out of it using these methods. The last specific method that I want to talk about is a slightly different one that's looking at this case of gene duplication. So in a system where a trait is being controlled by a gene, if you end up with that gene being duplicated, it increases the expression level and can change that trait. And so this has been found by this group led by Laszlo Nagy, where they were looking at white rot fungi, which can break down hardwood, as opposed to regular rot species, which can't. And they found that sort of, at the common ancestor of all of the white rot species in green, they found this massive gene duplication event. And subsequently, when there's been species within that clade that don't have white rot, they've seen that there's been a subsequent loss of the large number of additional copies of the gene. So if you sort of have a species where you've got some trait that you see only really arises in one particular clade, and you see the sort of repeated loss pattern of that trait, then this method can be good for trying to identify if it may have been a gene duplication event that drove that change in phenotype. So to wrap things up, I just want to give a quick overview of what the workflow looks like for PhyloG2P analysis. So on the left here is a reminder of that first figure, where now you sort of hopefully have a bit of a sense of what it looks like when we're doing this analysis on these deeper parts of the tree where we're using multiple species and their relationships to try and identify traits. And so to run PhyloG2P analysis, you'll need genomic sequences, so large number of genes in an alignment for the species you're interested in. You can also use other omics data for some of these methods. They have the capacity to add in transcriptomics and things like DNA methylation signals and other bits of genomic data. And you also need trait data to understand sort of where the replication that's occurred. And again, there's been a lot of evidence to suggest that having good mechanistic understandings of the traits that you're looking at and understanding sort of how they vary within species can really assist in being able to get the most out of this analysis. And so I think that the marrying of these two things makes it very fit for the center, but we have a lot of people with a lot of expertise in particular species in the sort of physiology space. And we also have people who have done a lot of work in the genomic space as well. And so, yeah, this is sort of a cool way to bring those two fields together to try and get the most out of both of them. Um, so to actually run these methods, you'll sort of put this into a PhyloG2P program that will typically need to be run on a high-powered computer because we're talking about big genomic sequences and a lot of processing that goes on. Sometimes this will need other parameters as well, but it really depends on the model, and selecting the model that you want to use is really like, in a GWAS analysis, there's sort of like a one size fits all to an extent. Whereas for these, as we said, there's multiple different ways that you can try and sort of look for replicated changes in genes. And so knowing which one to use or using all of them requires a bit of sort of thought and care around the system that you're using. And so out of this, you'll get a list of candidate genes with sort of a ranking score. So this is similar to how like a Manhattan plot would work, where you've got some genes that you're very confident are related to it and other ones that you're less confident. And so these still require sort of functional follow-up in the same way that GWAS does, where you'll need to do knockouts or mutant studies. But yeah, that's the sort of the general workflow. And so to wrap things up, I'd like to mention again that I'm writing this review paper that'll hopefully be published sometime next year. And I'm also developing PhyloG2P methods of my own for my PhD that are focused on taking better advantage of full genome alignments. And I'd finally like to thank my supervisors for their help with the review paper and with this presentation.
Yeah, thanks, Arlie. Very good overview.